The origin of our limbs takes us back 400 million years. At that time, life on our planet was exclusively aquatic. This was the moment when boned fish, which make up the most of today's species, diverged from cartilaginous fish, such as sharks and rays. These conserved anatomically primitive characteristics, especially a series of flaps of skin which protected their gills. These flaps were stiffened by arcs of cartilage, which could be related to our limbs. An astonishing hypothesis, tested by Professor Andrew Gillis. To undertake his research on the subject, Andrew regularly sets up at the Marine Biology Laboratory of Woods Hole, near Boston. On this Atlantic coast, specimens are right at hand, and every morning Andrew can expect a delivery of fresh fish. As the shark is a cartilaginous fish that's difficult to handle, Andrew studies the ray, more common and easy to capture.
The anatomical features that we are particularly interested in are the features of the gills in cartilaginous fishes. And so one of the things that's really interesting about their gills is that they have little cartilaginous rods that extend off of their gills. And these features are particularly interesting because it was proposed over 150 years ago that they could represent sort of an evolutionary precursor to limbs. And so just as we have a limb that extends off of a shoulder, the gills of cartilaginous fishes have little rods called branchial rays that extend off a gill cartilage at the side of the head.
To observe this anatomical peculiarity, Andrew Gillis needs to identify the skeletal structure of rays. That's why he programs X-rays for the freshly delivered fish. A light anesthetic bath is needed so that the subject doesn't tremble during the X-ray. While the animal's skeletal structure appears clearly, the gill arches don't stand out with the detail expected.
You got the whole gill region for sure, but we just can't really see. I think the gill elements may just be too small and too lightly mineralized to see with this imaging method.
To be sure not to miss any clue, Andrew also asks for a ventral image of the rays. The gill arches appear more clearly, but the relationship of the structure with our skeleton appears distant. The link is perhaps at a less obvious level. It is in ray embryos that Andrew will seek the origin of our limbs. He samples specimens delivered by the morning boat.
Embryonic development has classically been viewed as a really key source of information in understanding the evolutionary relationships among organisms and also among structures within an organism. And so it's generally thought that structures that share an evolutionary history will share common developmental programs.
Andrew opens the protective coating of the embryos at different stages of their development. Then he can observe how the gill arches form through the microscope.
Much like the development of limbs in mammals, the gills of cartilaginous fishes begin to form as a little bud that forms off the side of the head, and it very much parallels the way that the limbs form in animals like ourselves.
This similarity isn't by chance, but is due to the action of the genes we share with the rays.
The genes that drive development of the human embryo and other vertebrate embryos are the same genes. These are homeobox genes. And these Hox genes, as they're called, create segmentation in the animal. And essentially, a limb is just a specialized segment of the vertebrate's segmented body. These particular genes are activated such that a limb develops out from the spinal cord at that level of development.
So it's the same genetic program that triggers the development of our limbs and the formation of gill arches in rays.
You can think of how, evolution solves problems not necessarily by inventing new ways to do things every single time, but perhaps by taking genetic programs that work perfectly well and reusing them again, maybe with some modification. And the similarities between the gills of cartilaginous fishes and the limbs of other jawed vertebrates could be an example of this reuse or recycling of a very ancient genetic program. Our work is focused really on the role of a particular gene called sonic hedgehog in the development of the gills of cartilaginous fishes. And this gene is known to be very important for the development of limbs, where it plays two very distinct roles. Initially, it's involved in telling the limb which way is front and which way is back, or which end will form a thumb and which end will form a pinky. And it's also involved in controlling the number of elements that form at the end of the limb, so the number of digits. And we've been looking at how the sonic hedgehog gene functions during the development of gills and branchial rays in cartilaginous fishes, and we've found that it functions actually in a very similar way.
To prove this, Andrew experimented with blocking this genetic program in the ray embryos.
So here we can see a skeleton of a skate gill. And in this normal gill, we can see that they have a set of these finger-like rods sticking off the gill skeleton. Now, when we treat the embryos with a drug that blocks the activity of the sonic hedgehog gene, what we can see is a really striking reduction in the number of these rods. So it's kind of like the equivalent of losing several fingers from your hand. This effect of the sonic hedgehog gene controlling the number of skeletal elements and of symmetry is something that seems to be shared between the sharks and skates and the humans.